Hi, I'm Dr. Joseph Humphreys, and today I'll be talking about the genetics of autism. First of all, I want to thank you for joining our webinar, and I'm very grateful for the opportunity to speak to you of the new research and the opportunities that not only you as parents of children with autism, as well as those who do not have children on the autistic spectrum, to learn more and to become more empowered with how you can help your children. First and foremost, the most important thing to know is that I want you to join my campaign. And the fact is that 1 in 6 children now and here in the United States are suffering from a developmental disorder. And we are committed at AskDrJoseph.com, which is my personal website, and Healing Solutions Pediatric Centers, which is the clinic that I work at spreading information on how, number one, we can prevent these developmental disorders, and number two, how we can treat them effectively using many means, medications to natural supplements to dietary changes. And that's really what the goal of this program is. So today we'll talk a lot about what the old model of the genetics of autism is, and how the paradigm is changing. And what I want to do is provide a neutral ground to educate all of the parents concerning both preventative and treatment options that revolve around the genetics of autism. So first of all, the old model of genetics states that genes lead to abnormal brain chemistry, that somehow there is a gene that creates behavior, and that this abnormal brain chemistry leads to this abnormal behavior, which is classified as autism, where we have the repetitive behaviors, the communication aspects, the social aspects. And thus why lots of research has been trying to find the gene that causes autism. However, the old model has yielded very little results, and actually really what I should say is too many results. And here, right here, are the chromosomes 1 through 22, and then including the sex chromosomes X and Y. And these little red dots are genes that have been implicated in autism, where a study has come out saying, hey, we found the genes in autism. We know what causes it. And every year to 2 years, a new study comes out saying, we found this one, we found that one. Currently, right now, the biggest model is in chromosome 16, a gene on chromosome 16. And this is a study that was done on Nature's Review in Genetics. However, there's been no studies really that back these results over and over and over again. So here is a compilation of all of the genetic studies, and what I want to show you is above this red line right here. Anything above it is a gene that has been what's known as statistically significant finding in autism, meaning that the research has shown that this gene is present in a very large proportion of autistic children, and we believe that may be a contributor or a cause to autism. Now below this red line is all these squares, circles, triangles. These are all the studies that say, hey, this is what gene we think it is, this is what gene we think it is, but you can see it does not cross the threshold of this statistically significant. They are usually suggestive, or hey, we think it is, but we're not so sure. And so really, there's only particularly 3 genes here that have consistently said, we find them very, very often. And they don't even get into the highly significant. It's very weak results. These are 7 genomic scans of the whole genome in autistic children to trying to find what may be the autism gene. Again, following this older model that a gene produces some abnormal brain chemistry that then contributes to the symptoms of autism. This model really is also based off of twin studies, and what they did, and here you can see here are the twin studies: Folstein and Rutter, Ritvo, Stevenberg, and then looking at different case reports. And what they did is they took monozygotic twins, meaning these are the identical twins. And here is how many identical twins, the 11, 23, 11, 16, that they found, and they looked at if one child had autism, what's the likelihood of that identical twin having autism? Well, this first study said only about 36% would. This study said 96, this said 91, this said 69, this one said 75%. So you can see it's all over the map. Then they looked at dizygotic, meaning they're twins, but they're not identical. They don't share the same genetic sequences. And so here, this was 0%, 24, so a lot lower percentage. So this is where they said, oh my goodness, look, it's so much different than dizygotic twins, monozygotic. We know there's a gene causing autism. Well, you can see here when we kind of put this all together, it's roughly about 70%. So if one child, an identical twin, had autism, it's about 70% likelihood that the twin would have autism, which is a very large number. However, when you looked at the studies from a symptomatic aspect, you have what's known as penetrance. And none of them found that in monozygotic twins they had a very strong penetrance, which means that the severity of autism widely varied. So even though one child may have autism, a twin, the second may have autism, but the symptoms are drastically less severe, or vice versa. And so genetically, when you have a gene that causes one specific abnormality or brain chemistry, you don't normally see a weak penetrance. This is the same thing for like your eye colors. Your eye colors can mix and match. Your dad may be blue, your mom may be brown, and you could come out with hazels, and that's kind of the example. So it's the old models have really been refuted that a one gene is causing a autistic spectrum. If it was just one particular gene, it would be the same thing over and over. You know, trisomy 21 is an example. You have trisomy 21, you have Down syndrome. There's very, very specific characteristics that fit Down syndrome. Yes, there is some change in severity, but it's very much different in the autism world. So this comes from the Mendelian and Darwin genetic theory, the old model too, that that our genes are passed on, and that creates, you know, evolution. Well, the genetic understanding that's based off of Darwin and Mendelian, it does not support also what's known as episodic bursts of inheritance, meaning that the rate of inheritance of a gene is usually equal, or a mutation is usually equal. So in 1970, the rate of Down syndrome is pretty much the same as the rate of Down syndrome today. But we don't find that in autism. We find that the rate of autism in the last 20 years has significantly increased higher and higher and higher. Now, when we have what's known as episodic bursts, or the increase in incidence of a disease, if it is genetically based, the only way that it can explain is what's known as mutations, meaning something has changed the genetic sequences. And mutations occur through two ways. They occur through either environmental pressures or through what's known as abnormal chromosome recombination. So here on the bottom right hand screen you can see this is a chromosome, and it comes together in what's known as metaphase as the eggs are dividing and the genetic code is dividing, and they come together and they bind, and then they start to separate out each way. They divide, and sometimes this little piece here can grab a snippet here, and this little piece can grab that piece, and so it becomes an unequal distribution of genetic material. So those are the only two ways that mutations can occur. And again, mutations are really the way to explain episodic bursts of inheritance if it's a genetic problem. So the California data really is the most best tracked data out there. They have some of the best databases out there for tracking autism rates, and you can see here mental diseases, epilepsy, cerebral palsy, which we know have a genetic component, have been pretty stable throughout the years. It's pretty a flat line, but here we have the autism rate going higher and higher and higher. Now, if we want to say that this is a genetic-based disease, the only way to accomplish that is by debunking the rise in autism. That they say, nope, there couldn't be a rise, it's not a rise, we're overdiagnosing, we're going to change the criteria. And that happened from around 2000 to around 2006-07, where the genetic burst or the epidemic was very, very highly argued that it was not occurring. Because again, if we did have a rise, this quick burst, we cannot explain it with our known knowledge of genetics. And so it would point to a different etiology, whether an environment or some type of abnormal chromosome. And so that was one of the reasons of why the argument of that this cannot be an autism epidemic is because, again, it was based off the old genetic model. So the new emerging model is changing the way we look at it. So again, the old model is we have a gene that makes abnormal brain patterns that then governs behavior. This means it's strongly genetic. It's governed by genes. The new argument, and what we're finding research pointing over and over and over again, is what's known as genetically influenced. So we have a gene that interacts with its environment. This is called epigenetics. That gene and environment combine to create an altered molecular or some type of cellular mechanism, which leads to altered tissue metabolism or altered connectivity and processing, giving us behavior, cognition, sensory motor, and somatic symptoms. So this is an emerging model that we have genes that are present, but it's the interaction that they have with the environment that then creates the behavior, or the observable phenotype. So in that model, we can say, well, heck, let's get rid of the environment, and maybe we can solve these aspects. And that's where we're heading today, is looking at what's the environment that is influencing either the genetic pattern or creating a abnormal genetics through what we know as mutations. So this emerging model supports what's known as polymorphisms, or single nucleotide polymorphisms. These are duplications or deletions of small strands of DNA that influence protein and enzyme structure. So that these abnormalities create a susceptible pathway that's then manipulated by the environment. So this is known as epigenetics. The environment influences our genes and how they react, and our genes is therefore presenting enzymes that are structurally different than the normal population. So an example of polymorphism is we have the genetic sequence GTTACA-TTAC, and this gives us an enzyme that has about 90% activity. Then we have a gene that's it's the same gene, GTTACA-TA, and then the one base pair is AAC. And what that does is it creates this enzyme activity that's only 75% of the normal value. And so this is called the kinetics of the enzyme being modified. So let's say that a child is born with this polymorphism. Not a problem. It's only a 25% drop in the enzyme activity. But then let's add the environment that says, I need 90% of that activity. So the body can't make up for that extra 25%, and thus we see abnormal biochemistry being influenced by a small little gene that's normally there, but the environment makes it an abnormal process. So the newest SNP that they're finding is on chromosome 16. This is published in the New England Journal of Medicine. Its association with a microdeletion and microduplication at chromosome 6, arm 11, region 2. And this was very big. Again, it came out and the news said, we found the autism gene. We know what's wrong. We know what's going on. And ran through the whole news, and everybody thought it was the greatest and biggest thing until we actually started to look at it. Now here's what's known as FISH. It's in situ hybridization. We take a little fluorescent model and we take the genetic codes, this is the chromosomes, and we try and match those. They kind of like Velcro stick to these sequences, and so you can see them here and here and here and here and here. Here's some more. And that's how we find these sequences. We find it and then we try and isolate it and see exactly what it is. So here's directly from the study. It showed that 1% of autistic children have a recurrent microdeletion or reciprocal microduplication at this region. Now, 1%, wow, that's not very big, right? We have 99% of autistic children not having this gene. But wait, the news was all over saying this is the autism gene. We found it. We know what it is. But yet only 1% carries this gene. Well, the difference is in a normal control, it's about 0.001%. So this gene is very, very, very highly more prevalent in the autistic world, But compared to controls, but in autism, it's actually very rare. So I didn't get very excited about this gene really at all. It was only a very small percentage of autistic children had this abnormality. Now what's important is this statement right here, showing that both parents were negative for this copy variant, suggesting the possibility of a mutation. We observed no deletions in the parents. So this is saying the parents didn't pass this gene on, which flies in the face of the old model of Mendelian and Darwin and Kaufman genetics, that it's not being passed on to mom. It's a mutation. It's a change in the genetic code from mom to dad. What they also found that this deletion does not cause severe autism in every case. So again, low penetrance. It wasn't, if the child has it, does not denote severity. It just said, hey, we have a higher chance of being on the autistic spectrum. So I didn't get really excited about this study. And also, this study didn't tell me what we can do about it. But this is again where the old model is still focusing on. Let's find one gene and let's say that's the gene that causes autism, and then we can go from there and find a cure for that one gene. It's just not happening. What was funny too is that they looked for other genes. Orexin 1 was a gene that was very, very, very highly touted as the autism gene. And in their data showed that they didn't see really any deletions or duplications of this Orexin 1. So they basically said that the data shows little evidence of a similar large or highly recurrent regions of these abnormalities outside of the chromosome 16. So as I said, they refuted all the other genetic research saying it's this gene, it's that gene, it's this gene. They said, nope, it's not. It's this one. It's chromosome 16. They don't have anything else. So again, thus the conundrum and why science is saying, golly, we can't find it. It's just, over and over again. It's because they're still focused on the old model. So the new model is looking at the polymorphisms. Now these three polymorphisms have over and over and over again been shown to be statistically significant, found in a large proportion of autism. These two polymorphisms are common, but they're not extremely highly significant. But COMT stands for catecholaminomethyltransferase, and here's the variant that they find. REF is the reduced folate carrier, and here's the gene, the sequence. And this is the transcobalamin, MTHFR, methylene tetrahydrofolate reductase, and GST, glutathione transferase. So not only were these polymorphisms found very highly in children on the autistic spectrum, they also were verified by what's known as the abnormalities produced by their precursors and byproducts. So this COMT, this enzyme, if it's not working, you're going to get a buildup of the products before it and a decrease in the byproducts after it, right? So we have a product going into the COMT enzyme, and the COMT enzyme changes it and gives a byproduct. Well, if the COMT enzyme is found to be abnormal, we should find a high precursors and low byproducts, which is exactly what they found on all of these genes. So not only do we find the genes in the autism world, we find that the precursors and byproducts were abnormal. So again, another thing, another backup saying these are important. We know they're abnormal. We know they're causing a problem. So when we take those polymorphisms, we look at what do they do? Well, the thing that we've got to understand is methylation. So methylation is the transfer of a methyl group, or carbon and hydrogen 3, which determines whether a gene is active or suppressed. And environmental exposures cause these polymorphisms to turn genes on and off or make them less active, which is what we've talked about, the environment influencing genetic activation or enzyme activity. Again, epigenetics, this is where we're going. And the core of methylation is the folic acid cycle. Well, if we remember right here, the reduced folate carrier is an abnormal problem. We have an abnormal folic acid cycle. Therefore, we have abnormal methylation. So genes are going to be turned on when they shouldn't be, and genes are going to be turned off when they should be on. So here's this cycle. It's very complex, I know, but here's this folate carrier, and here's the cell, And outside we can measure serum folate levels, and inside we can look at this 5-methyl tetrahydrofolate right here. And here's these genes that I talked about: MTHFR, methionine synthase, folate carrier. All of these genes that I've talked about play in this methylation cycle. And these sequences have over and over and over again been implicated and found in the autism world. So again, here's that reductase folate carrier. If this is abnormal, serum folate cannot get into a cell and therefore run this methylation cycle. So the reduced folate carrier delivers folates into the cell. So if this is abnormal, children are 2 times more likely to develop autism with this polymorphism. And what we find in my history is I find a history of birth defects in siblings and even in the child themselves. I see elevated blood folate levels that I can test children for. I also see in history multiple miscarriages in mother or in sisters or in grandmothers. I can't tell you how many times I've done an intake with a mother that has a child on the autistic spectrum, and they had 4 or 5 miscarriages and they had a difficult time getting pregnant, and finally, after many years of trying to get pregnant, they got pregnant, and then lo and behold, they were devastated by an autism diagnosis. And multiple times when I've talked to parents and say, oh, we've had a history of miscarriages, I've tested the child and we've found there was a folate carrier problem in the child. And then I test mom and I find that exact same abnormality in mom. And so it's very specific. We can find these things not only through history but through testing. Another one is the transcobalamin 2. So this is a carrier protein, transcobalamin 2. And what happens is this is our GI tract over here. Cobalamin, we eat it, is carried by intrinsic factor. It binds to it, and it's absorbed in our gut. This is the brush border in our stomach, in our intestinal lining. And then we have a series of process of enzyme degradation to where cobalamin is released, and transcobalamin 2, it's a protein, binds. And the only way that cobalamin, which is another form of vitamin B12, is released into our blood and then later taking up into other cells is if transcobalamin 2 binding protein is there. If we don't have transcobalamin 2, we do not release cobalamin or vitamin B12. So again, this is a transport carrier that's required for B12 to be uptaked into the cell and released from our GI. So if we have a polymorphism in this abnormality, there's a decreased binding affinity of the transcobalamin 2 protein. Therefore, there's a 1.7-fold increase in the risk of developing autism. This is very common associated with it, and what we find is where we have normal development up to a 16, 18-month development period, and then we experience severe regression. We often find in the history Alzheimer's in the family history. We see periods of anemia, fatigue, lack of weight gain. Kids will come in and their growth is really low. Blood-wise, we see high methylmalonic acid. We see low immunity. They have chronic ear infections. They've had a lot of upper respiratory abnormalities, which leads to low levels of gamma globulins. These are the immunoglobulins that keep us sick. We also see infant vomiting. Sometimes these kids that have extreme colic is due to a transcobalamin problem where they can't absorb vitamin B12. So again, here's that methylation cycle, and we can see here's where B12 works. So if we're having abnormals with B12, we're not going to run that methylation cycle. Very important to know. So here's the COMT polymorphism, the catecholaminomethyltransferase, and this has to do with our neurotransmitters. So when we eat our food, we break down food to basic amino acids, and we take the amino acid tyrosine. Tyrosine is created to L-DOPA. L-DOPA is then created dopamine. Dopamine is so very important for attention, focus, concentration. Dopamine is converted to norepinephrine and epinephrine. These are our flight or fight hormones that when we get scared, we get high levels of norepinephrine and epinephrine. Well, here's that COMT. It's responsible for breaking down the dopamine or norepinephrine and epinephrine. When we get these levels high, well, we need to get rid of them. We need to pull them down. And these lead to the byproducts of homovanillic acid, vanillomandelic acid, metanephrine. And what we find is we find kids with COMT abnormalities. They get either it can be extremely fast, so it breaks down dopamine very quickly, giving high levels of homovanillic acid. And this causes lack of attention, lack of focus, or it can be abnormal, which gives a high level of dopamine. This is associated with extreme aggression. Kids that are diagnosed schizophrenic and bipolar have abnormal dopamine levels. A lot of the medications used to control behavior, such as risperidone, modifies our dopamine level. Well, heck, if they would just look at the COMT polymorphisms, you would find that we can modulate dopamine by normalizing COMT. Same thing with norepinephrine and epinephrine. When kids have these COMT polymorphisms, they don't break down these fight or flight hormones. So it's almost like they're living on the edge. Any little tiny push sends these epinephrine levels already skyrocketing. They're already elevated, so any little tiny thing just pushes them over the side. So this is what I hear from parents saying, it's like I'm always walking on eggshells. I never know what's going to set them off. And when it sets them off, it's a 2 to 3 hour tantrum. These kids don't know how to come down because these epinephrine levels stay up elevated all the time. So again, here's those COMT enzymes. So the catecholamine hormones are methylated, and it helps them in the degradation process. Again, we've talked about dopamine's important for attention, execution of cognition. When you combine also this with like the transcobalamin abnormality, polymorphism, it increases the risk of autism by 7-fold. So we are finding that these polymorphisms, which are in the genetic sequences that are normally there, we get combinations, and we get an environmental stress that says we need more or we need to break it down quicker, raises these levels extremely. So again, what we see is we see hyperactivity, we see a thyroid problem, ADHD, and then I can look in the blood and I can look at these breakdown products very easily. We see hormonal fluctuations, and as I said, schizophrenia is very common. So methylene tetrahydrofolate reductase, as I said, this is one that's kind of marginally significant. It's not, we haven't found it consistently, but it's one that I do find. And it converts folic acid to the active form of 5-methyl tetrahydrofolate. It's again using that methylation cycle. We find polymorphisms in this enzyme. It decreases the activity by up to 67%. What we find is a high incidence of inflammatory bowel disease. I see also high levels of psychiatric diseases like depression in families. Again, this is also one I see with miscarriages. And urinary, I see high what's known as forminoglutamic acid or high FIGLU. So when this is combined with the RFC polymorphism, it increases the rate of autism by 3. It's just amazing what we can see over and over again, identify what we know. You know, we should be testing moms and we should be testing kids at birth to see, hey, they have these polymorphisms. We need to change our environment then so that we don't stress these systems. So here's this MTHFR. It takes methionine and converts it into homocysteine, methyl tetrahydrofolate reductase. Here it takes the folic acid we ate, we methylate it, and that's used in the methionine synthase enzyme, which then creates homocysteine into SAH, which is the methyldonal for methylation. So again, here's this transcobalamin where it's used. Here's the RFC, here's the MTHFR. You can see it's all centered in this methylation cycle. Down here is also what a lot of times we find abnormal, and if we have this system's not running from methionine and homocysteine because we have one of these problems, we get abnormal levels in cystathione, which is a precursor to glutathione, which we know is a major antioxidant. So here's the MTHFR frequency. This is done by Dr. James. Here's the different sequences we can find in the MTHFR. And we can see in the autistic children, here's the percentage that have this polymorphism, and they are statistically significant. Anything that's 0.05 or less is statistically significant that we find, and it reduces these enzyme frequencies. It's found very common in autistic children. The GST1 is the glutathione recycling, and glutathione is a tripeptide or 3 peptides of cysteine, glycine, and glutamine. And glutathione regulates nitrogen and oxygen free radical scavengers. It's the free radicals damage our cells. And they damage our body, and it causes systems to break down. It maintains our cell membrane integrity. It helps us detox things. You know, a lot of the concepts of detoxification falls into this, and it helps us with apoptosis, meaning if a cell's abnormal, our body wants to kill it and get rid of it. Well, if it doesn't have glutathione, it's not going to kill it, and therefore that cell that's damaged reproduces on and on and on. That's the concept of cancer. So GST1 helps recycle used glutathione. So again, if we have GST1 and combine it with RFC, we have a 2.7-fold increase in autism. And what we find when we have a GST1 null is that we have smell sensitivities. They smell things, they sniff things, they take their fingers, they put their hands down their pants and then bring it and smell in their fingers. They have brittle nails, abnormal skin patches. We see that these kids have extra metals in their bodies. We see normal exposure built up. Increased what's known as GSSH, this is oxidized glutathione I find in their blood, or decreased serum cysteine. These are things I can find that tell me that this enzyme system is abnormal. So here's a study showing glutathione in autistic compared to control child. You can see intracellular glutathione levels extremely low in the autistic world. And in autism, the free radical generation, at baseline, we already have tons of free radicals compared to control. When we stimulate them to produce more, they just produce more free radicals than the controls. We know kids on the autistic spectrum have glutathione problems. So what can you do? What's the point of this talk? And the point really is we have options that point to genetic sequences, and we have options that we can manipulate these sequences by changing the environment. And a lot of people are doing genetic screening and say, I want to know if my child has all these screens. A lot of these are not commercially available. We can screen them, and that's no problem. But oftentimes, I don't recommend doing it. And what I do is I look at the byproducts and the precursors, and it helps me say, yep, I know that sequence is slower than normal, so let's focus on that. And that's what's known as a therapeutic trial, where it takes me, it costs parents $1,000 to do a genetic screening of these polymorphisms. It takes us $50 to do a therapeutic trial of a small little supplement sometimes, and we see changes, and I can test the byproducts. So I can do both. I do either one. Sometimes I do test them, but oftentimes I'm more likely to say, let's do a therapeutic trial. But we do have polymorphisms we know we can change. Blood tests do often tell us the byproducts, and I do that very often, and insurance usually does cover that. And lastly, work on the body burden, meaning changing the environment. So just an example, here's a panel I run. This is run on a 7-year-old female who has autistic spectrum. She falls really more on the Asperger scale, though, when I evaluate her. And again, here, like talked about, the vanillin, benzoylic acid, homovanillic acid, these are pretty normal. Anything in the green is normal. 5-hydroxyindoleacetic acid, this is elevated. This is a serotonin problem. So it's a neurotransmitter. This is often another one that's hasn't been published much, but that we're looking at. But here's the big one, her methylmalonic acid. This is that transcobalamin problem and not processing B12. And we see it's elevated. It's in this kind of red level. We really want it below the 2.2. And her behavior fit it. And so what I did is I treated this abnormality by bypassing, so the body again takes cobalamin, it has to methylate it and activate it to methyl transcobalamin. So what I do is I just bypass the precursor and give the byproduct, so it bypasses that gene that we have normal. And within a matter of about 6 weeks, you can see this methylmalonic acid falls down into a normal level. This is what I tested afterwards. Falls into a normal level of 1.1. We had significant gains in language and sensory function and communication and becoming more aware of the environment. So this is what I like to do and how I really focus on looking at the genetic sequences in the autism. So here's again that transcobalamin at B12 comes into this process and runs and activates, help with methionine synthase, taking methionine to SAMe, which normalizes the methylation cycle. Methylation cycle, again, as I've talked about, methylation turns genes on and off. It helps things run smoothly, particularly with attention and focus, dealing with the dopamine receptors. And so when this was abnormal, we're not using B12, I found the methionine levels were abnormal, homocysteine levels were abnormal. When I corrected it by giving the right active form of B12, this methionine synthase enzyme started running again. It started running this little cycle here, which gave me normal methylation cycle, and it ran over this process, which gave me the normal folate to use over for other synthesis processes. So the body burden, this is the environment that I talk about. We want to reduce the environment burden on our children. This was a phenomenal study by the Environmental Working Group talking about pollutions in newborn. And what they did is brand new babies, after they are born, within 2 or 3 minutes, the cord blood, so the blood coming straight out of the umbilical cord. Now, this is not mom's blood. Mom's blood really doesn't mix with baby's blood very much. They have these little villi, and they transport nutrients across the villi and into the umbilical cord. And so they said, well, we just want to know what the child's burden is, and so some from the cord blood. And what they found is they found 287 chemicals that they detected. 180 were known to cause cancer in humans or animals. 217 were toxic to the brain and nervous system. And 208 caused birth defects or abnormal development in animal testing. So really, there are no studies that show the dangers of pre or even postnatal exposure with this complex mixture of carcinogens, developmental toxins, and neurotoxins. They've never been studied. So these levels were very different than what we know. That one dose of this we know at this level causes problems. Well, these were below that. However, when you combine it with 287 other chemicals, we know it causes problems. We know it can change environment. As we've showed you for in the studies, parents weren't having the same genes that the kids had. They were mutations. Could this be the source of the mutations? I think it can be. So this is another area that I often discuss with parents is, hey, before you get pregnant, let's help moms get as healthy as we can. Let's reduce their toxic burden. Let's look at the genetic sequences. Let's get them healthy, diet changes, supplements, whatever we need to do to reduce our chances of having a child with a developmental disorder. So I wanted to thank you for joining this webinar. If you enjoyed it, please pass this conference talk on. It's free for anybody out there. If you'd like to have more information and receive our monthly newsletter of our webinars, as well as our I publish new research and opportunity to join forums to talk with parents and options and solutions, you can join that at AskDrJoseph.com. And lastly, to remember that there is always hope. No matter where your child is, I believe we can make gains and strides and functions. And this is from a personal experience with family members, with patients. I've been doing this since 2004. I know that children in the autistic spectrum can be recovered. And I know we can make great gains. And we've just got to focus, and we've got to find core issues, and we've got to look beyond sometimes traditional medicine and use it when we need to, and look at all of our options that we have and treat these children as individuals and not cookie cutter boxes. I sure very much appreciate the opportunity to interact with your children and to have the pleasure and honor to help you on that pathway. Should you have any questions, please feel free to contact me at AskDrJoseph.com or at our clinic, which are both listed on that site. Thank you, and have a good night.